webinar. So today I want to introduce TomoAnalysis, how it works, and what is the background or the running principles for the analysis. And then I'll briefly show how the workflow goes in TomoAnalysis. So let's see how the TomoAnalysis makes the analysis. So this is the typical data that acquired by the color tomography instruments from Tomocube. So basically it's three-dimensional and it's label-free, and it should be correlated with fluorescent signals to have better understanding about the sample. And at the same time, we can take time-lapse data. So we can say the data is very multimodal and very complex. So we need to make a dedicated or an analysis software that is comfortable to deal with this kind of data. And at the same time, we know that we have released a former president of the current HTX1 system, which is HT1H or 2H. So we need to deal with these kind of data as well. So we have made a new software based on these kind of requirements. So let's see what kind of analysis softwares are at the market. The first thing for microscope manufacturer is the first software to have is dedicated software for their own modalities or imaging techniques. So it should be optimized for specific imaging modalities and workflows. So in terms of when we say about our hologram data, we need to have some requirements to deal with those kind of data. And there are a bunch of things like light sheet microscopy and confocal microscopy, which is specialized for typical manufacturers. So for each categories of modalities, they have built a dedicated software to deal with those kind of things more conveniently. On the other hand, there is open source software, which is ImageJ or Napari Cell Profiler and so on. They are very well known for its flexible analysis because there are a variety of kind of images that is to be dealt with. So they made it very flexible to have more flexibility and more accessibility about the data characteristics. And because of the open structure, the contributors contribute many diverse techniques, very novel techniques. So you can access very novel techniques with that kind of open source software. So when it comes about the novelties or a new analysis techniques, it's very convenient to access those kind of things. And on the other side, there's very genuine programming languages. So some people can use Python, Java, C++ to have their own programs. The characteristic of this kind of software is it's very time-efficient analysis, very powerful for a very specific and repetitive task. So some people are dedicated on some very specific targets, then they build their own programs to have more suitable or efficient analysis. But in a normal, typical flexible analysis, it's very limited to use those kind of things for more general biologists. So we understand these kind of requirements and demands. So we developed TomoAnalysis to reflect these kind of requirements. So it's dedicated for holotomography, which is 3D. And some people want to analyze the data in 2D or in MIP. So we adopted techniques for 2Ds as well. So it can deal with 2D or 3D data in holotomography. And we incorporate modular approach for flexible analysis. So you can combine multiple or different kind of processes, which we call processes for each modules. So we can combine multiple processes to build your own or very specific to reflect a specific requirements or specific targets. And we also incorporate our proprietary codes for efficient analysis, which uses GPU to enhance the efficiency of analysis. So this is the basic idea how we developed TomoAnalysis. And let's see how bioimage analysis looks like. So this is the typical analysis procedure for quantitative analysis. So the important thing here is quantification. And for quantification, you need segmentation, which means to define the area or region of interest for your own analysis. And for this segmentation, you require pre-processing and post-processing to have a better refined definition of the region of interest. So for segmentation, the most well-known or the simplest form of segmentation is thresholding in fluorescence. So by selecting a certain value, which is called threshold value, when some of the parts of the image is higher than the threshold value, it is considered there is some targets. And when the value is smaller than threshold, we call it there's not, we think there is nothing. So by using the threshold, we can define the segmentation area, like in the middle. And we see small holes here, which is not desirable. Then we can get some kind of post-processing to fill these holes. So by using fill hole process, we can fill up those dark spots here and have these nicer boundaries or regions for the analysis and take the quantification for these regions. And before segmentation, we can also apply some kind of pre-processing like denoising or Gaussian filters and so on to have a nicer image for the segmentation. So this is the simplest form of the analysis procedure: pre-processing, segmentation, post-processing, and then quantification. So these processes are done in TomoAnalysis. So based on these basic steps, we have classified 3 strategies for segmentation. The first one is fluorescence-based tagging. So most biologists are familiar with fluorescence. In fluorescence, they usually use thresholding. So by using thresholding, they can take the area of the specific labels, and by using that, they can easily get the quantification results. On the other hand, when it comes to the holotomography, it's non-specific. So we should take some more criteria to get the uniform object. But the basic idea is very similar with fluorescence. So it uses holotomographic image, not the fluorescence image, but the idea is very similar. So in this case, we captures the unique RI features like the RI value or its gradient or its relative intensities, relative change across the region. So based on that features of the object, we can get the segmentation of the uniform object. The examples of like this, lipid drumlets or mitochondria. So we know that mitochondria have substructures, but in holotomography, the resolution is not enough to resolve those tiny structures. So in holotomography, it's rather imaged as a single, it's kind of strand. So it's quite uniform in terms of holotomography, and we take advantage of that uniformity to get the mitochondria and segmentation. And the other part is object with an internal structure like cells or nucleus. So the structures like cell or nucleus, because they have very complex structures inside the object, it's very difficult to get the segmentation with a few criteria. So it requires complex and context-based segmentation method. So that's the hardest part in segmentation. But there are a few ways to get those objects segmented. So we will briefly see those results as well. So this is how the segmentation happens in TomoAnalysis. On the left side, it's fluorescence tagged object. So in the left side, the nucleoli is labeled with a kind of fluorescence dye, and we can segment those areas based on the fluorescence signal and get some quantification results. And in the middle, you can see the lipid droplets segmented by TomoAnalysis. You can see bright dots here, whose result is displayed on below. And on the right side, it's cell segmentation. By capturing the Cs of the cells and expanding the Cs for each cell, we can get these kind of segmentation results and get some volume and other geometric information from these segmentation results. So these three strategies are incorporated in TomoAnalysis. And the good thing is these all segmentation is based on the tiny blocks of which is called processes. And by combining those processes, we can get these kind of segmentation pipelines, analysis pipelines. So it's very flexible. So by combining these processing, segmentation, post-processing, quantification, by combining these processes, we can get strategies for various targets. And the good thing is, by mixing or by combining those strategies, we can have more complex analysis, like multiple targets at the same time. And we also can do some relational analysis. For example, like when we segment or analyze lipid droplets, we can get the segmentation of LDs. But at the same time, we can segment those cells. And by combining the two segmentation results, we can get the LD quantification per cell. So by this way, we can extend the analysis scope for more complex ideas. So this is one of the examples of the combined analysis. So here on the left side, it's tagged with two different kind of fluorescence. One targets for the nucleus and the other for the nucleoli. And by segmenting those two organelles or the targets separately, we can get these masks. So one for the nucleus and the other for the nucleoli. And by mixing the two, we can get this kind of analysis. So here, 1, 2, 3 means the nucleus, and here, 1, 2, 3. And from this, we can see the average Li value of the nucleus is very similar, but the nucleoli value differs depending on the cells. So by this way, we can extend the knowledge about the targets using fluorescence and the holotomography. And the other analysis combination is using fluorescence and HT for like drug quantification, drug intake quantification. So on the left top side, it's fluorescence labeled drugs. We treated the cell with drugs and take the fluorescence image like this. So we can see that the drugs are inside the cell. And at the same time, we can get the segmentation of the cells. And by combining the two, we can get the drug intake per cell, depending using these two combination of these two analysis results. By this, we can get this kind of advanced analysis by combining the two strategies. So this is the example how the TomoAnalysis can be configured to have very diverse type of analysis. So for this kind of diverse analysis, we have incorporated a myriad of processes for pre-processing, for segmentation, for post-processing, and for quantification. So there are more than 100 processes at the moment, and we are adding processes as we need and as we as TA evolves. So once you use TA TomoAnalysis and find some features or processes are lacking, then just let us know to have those features or those processes for better analysis. And this is the pipelines that we have found useful for analysis. So one for fluorescence, so we can extend the channels from 1 to 2, and lipid drumlets, and instant segmentation for cells, and those fluorescence intensity measurement for the cells, and lipid droplet analysis for each cell. So by combining the three pipelines on top, we can extend this kind of advanced analysis based on those basic segmentation analysis pipeline. And the important thing here again is the quantification parameters. So because TomoAnalysis deals with 3D information, we give a geometric and physical information based on 3D. So the center point and the center of mass in 3D, and volume, surface area, projection area, sphericity, diameter, ferrite length, and so on. And for physical parameters, there's dryness, concentration, and the mean RI or max, mean, and range. Such kind of statistical information about the distribution of the RI value. So we can extend those geometric and physical values as we evolve the TA. So let us know if you want more parameters to add on your analysis. And after the analysis, it's important to have a visualization. So based on the analysis results, you can extend the analysis in 3D. So based on the segmentation, you can crop or the focus, or you can focus on the targets. Here we acquire the cell segmentation, and based on the results, we can get this kind of visualization in 3D. So that's the benefit of having 3D analysis. So based on the analysis result, we can have this kind of three-dimensional visual rendering as well as quantification results. So let's see how the TomoAnalysis works in reality. So the first thing that we should know about the TomoAnalysis is how the workflow goes. So in reality, we should select the files and select what kind of modalities that we will use. And when the data contains time-lapse data, we need to select the time point. So the second process is select the workset, which we call workset, which defines the channel and the time point for the analysis. And then we can set the region of interest if we want, based on for each workset to exclude or include some of the region of interest. And the first three steps are for the preparation of the data set. And from 4 to 5 is the actual analysis. So the step 4 is for parameter adjustment for optimal analysis. So when you select pipelines, analysis pipelines, there's some parameters to adjust for better analysis, and that's the step for preparing the analysis pipeline. And then you run in batch, because you can select multiple files and multiple worksets. The analysis software automatically runs the pipelines through the files or the worksets you have chosen. So that's the step for the batch analysis. And after the batch run ends, you can review the results. So this is how the workflow goes for the in TomoAnalysis. So based on that workflow, this is how the step 4 looks like. So when it comes to the segmentation of fluorescence type objects, the data looks like this when you load the data. And by simply selecting these threshold values, like number 2 here, actually for this fluorescence segmentation, it's very straightforward. And the only parameter, usually the only parameter that you need to control is the threshold value. So depending on the experiment, the intensity, fluorescence intensity can vary. So yeah, to reflect the intensity changes, you can adjust the threshold value like this in this second process. Here, I chose 250, for example, and get this kind of segmentation. And if you're satisfied with these settings, then you can set this value for the batch analysis, and you can go with these parameters. So this is example of how the analysis goes. So it's very straightforward. So here I intentionally set a value too high for these images. So you see that some of the nucleus here, actually it's nucleus, the nucleus is stained. So here you can see some of the nucleus is filled and there is no spots in the middle, but some of the areas looks very spotty. So we need to fill. Actually, I applied the fill hole process to fill the holes, but it's very sparse, so it couldn't cover the whole area except these two. So in this case, you need to adjust the parameters to reflect this kind of situation. So you may need, lower the value to include those excluded area here. So let's see how the process looks like for these fluorescence tagged objects. So here there are in total 5 processes. One for pre-processing. So this is Gaussian filter for smoothing. And the second part is thresholding, which is for segmentation. And the third part is filling holes, which is for post-processing. And the 4 and 5, it's for quantification. The fourth process is for labeling, which means after the thresholding, actually the objects are not separated one another. So by assigning labels for each object, we can get the quantification result per object. And so the fourth process does the job for labeling. And based on the label mask, number 5, analysis on target, which quantifies the values for that target. So in this 5th process, the result in RI or volume and other parts of the measurement data comes. So this is how the process for the fluorescence segmentation works. And when it comes to the uniform objects, it's very similar to fluorescence-based segmentation, but it's not done by a single threshold like fluorescence. So we combine 2 kind of segmentation criteria to have better segmentation results over these lipid droplets. So here we incorporate 2 different criteria. One is absolute RI value, which is here like 1.35. So we lowered a value a little lower than usual value to incorporate unusual LDs as well. And besides these relative RI value, we incorporated a relative RI value. So in some cases, cells, when cells condense, will cells contract, the relative the refractive RI value gets higher. So we wanted to exclude those unnecessary parts. So to avoid those kind of things, we applied this kind of relative RI value segment criteria. So in this case, we applied 180, which is actually, it should be divided to 10,000 to have RI value. So it corresponds to 0.018 in terms of RI. So those parts whose relative RI value is higher than 0.018 can be segmented with this kind of criteria. So by mixing the two, we can get these kind of ideas. So let's see how it looks like. So when those areas whose RI value is higher than 1.35, it's like this. So you can see these parts are segmented or distinct, segmented as LDs because it has high RI value. But we know that this is not the LDs, but some kind of contracted cell parts. So on top of this, we can apply this delta RI value, which means the RI value relative to its neighborhood, and whose value should be larger than 0.018. And in this case, we can get this kind of results, which is more like LDs. And by having the intersection of these two results, we can get nicer LD results like you showed before. So this is how the analysis process goes. Oh, yes. So here you see that the on the left side, it's the pipelines for the analysis of LDs. And on the right side, you can see the outputs in the middle. So the outputs produced by each process is listed here like this. So in total, including the source or the original data files, there are in total 7 data outputs for that is used for the analysis. And if you want, you can visit each layer or each output to see how the analysis went on during the process. And you can refine the results by visiting those outputs and controlling or adjust the parameters for each relevant process. And also you saw that the results can be displayed in 3D, and also the targets like the LDs can be separately displayed in 3D to have better idea about the analysis results. So this is how the analysis works in TomoAnalysis. And this is segmentation of object with internal structures, which is the segmentation result of cells. So I briefly introduced how it works here. So here you see the cells are segmented in different colors. And the way this is done is by expanding the seed, taking the seed. It's actually not the real seed of the cell, but it's mathematically calculated the seeds of each cells. And by extending the seeds to make it grow, so by expanding the seed, we can get these segmentation results. And after that, we can label those individual cells based on these seeds. So we can get this kind of good, great segmentation results from these pipelines. So this is the same. This is the procedure or the segmentation pipeline is made of those basic processes that we have implemented. So depending on the processes, you can do many kinds of analysis as you wish. And this is the screenshot of the object of these cells, and this is the same. And when you get the analysis results, you can do the same with the in 3D as well. So based on that segmentation results, you can get this kind of rendering in 3D and get more idea. And you can actually, separate those cells one by one based on the segmentation results. That's the benefit of the segmentation. So by mixing those techniques, you can have very diverse analysis. And you can do many more segmentation or analysis based on these strategies. So you can combine many basic strategies to have diverse ideas. So for example, you can get the LD or mitochondria analysis, and you can do the analysis based on cell as well, and you can combine the two to have better understanding. So in this case, you can use the whole tomograph data to get these kind of ideas. And at the same time, you can tagging the targets with fluorescence, and at the same time, you can get the instance of cells with HT. And the benefit of these strategy, fluorescence and fluorescence tagging and using the cell segmentation, is that you can save your fluorescence dyes. So when you use fluorescence, you should tag many things to have advanced analysis. So on top of the target cells, you target the fluorescent target that you are interested in. Other than that, you need to use diverse, many basic fluorescence to stain the cells or nucleus, because you need separation between the targets based on the cells. But when you use color tomography, you don't need that kind of extra stains. You can use holotomography to get the segmentation of cells or nucleus from holotomography and combine the result with the fluorescence data, so you can get more, you can get clearer fluorescence signal because you save the fluorescence. So that's the benefit of these combined strategies. And you can do the multiple fluorescence experiments and do the experiments or analysis based on the multiple fluorescence, as I shown before. So this is the segmentation strategies that can be done in TomoAnalysis. And you can ask some of the questions about TomoAnalysis. So I added here briefly. The most frequent questions are like these. So does TomoAnalysis handle data from 2H or 1H, which is the predecessor of the HTX1, which is the current instrument that we promote? And the answer is yes. The process in TA can be applied to any type of data, including TCF. So it does not distinguish between the data from 2H and the data from X1. So you can use the data from any instrument that Tomocube has made. And one of the examples that I shown before, the fluorescent data, it's acquired from 2H. So that's the example of the TA can handle that the data from the former 2H instruments. And the second question is, am I able to open the analysis results in other programs like ImageJ? So yes, the answer is yes. The analysis result in TomoAnalysis can be exported in TIFF and RAW formats when it comes to if the data is in image format like the holotomograph data and the label data. And the measurement data, which is in tabular format, it can be exported in CSV. So you can open the data in Excel or other spreadsheet for more statistical or more analysis. And third question is, does TomoAnalysis support machine learning or deep learning algorithm? So the answer for the time being is not now, but the AI functions will be added soon. So we are developing, incorporating those algorithms that is embedded as a process in TomoAnalysis. And the good thing is when we incorporate those functions, AI functions, we also thought about incorporating the functions to run third-party programs as a process. So it can provide functions to use other programs in the headless mode. So it can run ImageJ or SEP-Profiler or other Python codes as a process in TomoAnalysis. So when you see the function of AI in TomoAnalysis, you can also see those third-party programs learn as a process in TomoAnalysis to extend the functions for more advanced and more diverse analysis. So this is the three questions that I have made or prepared for frequent questions. And then I'll go into the real analysis based on the software. So I think I can get some questions from you if there is. So Liu Xuan asked if I wonder is there a specific RI value that you would suggest to segment mitochondria? So at the moment, the RI value, the simple RI value does not guarantee the segmentation of mitochondria. Although mitochondria have some specific RI value, the RI value is not specific to mitochondria. So when you use those simple RI value, then it'll also segment those other parts like cell membrane or the nucleoli as well. So we are searching for more better segmentation analysis, and at the moment we have two strategies for that. So mitochondria, the mitochondria when it is segmented, it's in a fiber. It's like fiber. So the shape of mitochondria can be used for the segmentation. And so by combining the features, the fiber-like feature, we can segment those mitochondria. And yeah, by taking those features as well, like LD, like LD, where the simple RI value does not guarantee the segmentation LD, and we added the relative RI values for the segmentation. For like that, for mitochondria, we can use the RI value for basic segmentation, but for better segmentation, we can apply the fiber-like features for the mitochondria. And actually, we are developing the feature, so we are not ready to release the mitochondrial segmentation pipelines at the moment. And at the same time, we are also thinking of a machine learning process for mitochondrial segmentation and other supervised learning algorithms. So we are successful in getting those mitochondrial segmentation using Elastic, and we are incorporating the features. As I said, we are incorporating machine learning features in TomoAnalysis, and we believe that you can also use machine learning features for segmenting mitochondria. In that case, you can, at the moment, you can use Elastic to run as a third-party program, but at the same time, we are aiming to give the function in TomoAnalysis, including the training procedures. So wait for the release of those features for the mitochondria analysis. And also, Mariana Kunze asked that if the fluorescence signal is only at the parameter of the organelle to segment, e.g. at the membrane, can the fluorescence tagged object workflow handle fluorescence of that sort, or does the fluorescence have to be present throughout the interior of the organelle? I'm asking because of the segmentation of yeast vacuoles. So there are two ways to get those kind of segmentation. So actually, it's not that difficult. So if we ask us, we can support. At the moment, we are very open to support your requirement because we are at the phase of developing pipelines. And I think it's very straightforward. So if the membrane is a fluorescence tagged, we can fill those regions. One way is to fill the regions based on the perimeter segmentation, like fill hole. And the other way is using a bit advanced process, which is called watershed. So based on the shape of the segmentation result, we can calculate the inside of the perimeter, and based on that, we can segment, or we can make a better segmentation mask for that vacuole. So I think we can try multiple ways to get those segmentation. And Hussein Kadem asked that my TA software has AI module, and by upgrading it, I will. It's just first.
Sorry, but could you, because it's Songshik, they raised their hand, so.
Okay.
So Songshik, can I? So we cannot hear you. So could you turn on the microphone?
Oh yeah. Okay. I don't think I can make the something can talk. How can I make him speak?
Yeah, Ahram, please just move on to the Hussein's another question. So this about the lipid drum analysis.
Okay. So there's Suchin Yadev asking, how should I approach the segmentation of nucleoli and quantify changes in nuclear reflective index when the threshold of certain drug-treated cell undergoes alteration? So the most difficult segmentation targets at the moment is nucleus and nucleoli, because the characteristics of nucleus and nuclei is very very special, because in living cells the RI value of nucleus is lower than the cytoplasm, so it's like void in the middle of the cell in terms of holotomeric data. So we've tried many ways to segment nucleus and nuclei, but yet we have not been successful. So that's the painful part for us. But when we have those machine learning features, then we think we would be able to segment those nucleus and nuclei. Actually, nuclei is, I think it's easier than nucleus, because when we get the nucleus segmentation, nuclei is just a bright part inside the nucleus. So the most important part here to segment nuclei is to have nucleus segmentation. So if we solve the problem for a nucleus, then the nuclei will be solved naturally. And Hussein Kadam asked does the software provide the number of LDs per cell? Yes. When we segment LDs, the LD droplets are separate by the droplets. But the thing here is that when the lipid droplet gets smaller and gets aggregated, it's very difficult to separate those aggregated lipid droplets. And it's the same, it's the situation is very similar in fluorescence. When LDs are stained with fluorescence and the LDs are too small or too close to one another, then it's very difficult to determine each lipid droplets because the signals are mixed, and it's very difficult to tell the boundaries of each droplet. So in our process, we neglect those small droplets because sometimes the lipid droplets can be confused with other small entities in cells. So it depends on your choice, but in the default pipelines that we provide at the moment, filters out those small LDs, which is usually smaller than 0.2 or 3 micrometer. So when you open the pipelines, you can change the settings for your own purpose, but I think it should be enough. And yes, the answer again, if I answer again, you can calculate the number of LDs, and you can make the distribution chart based on the numbers and the size of the labeled LDs. And I wonder, oh yeah, someone already told, so if I address it again, my TA software has AI module. By upgrading it, I will lose this feature. I mean, the new software is only based on RI segmentation. So at the moment, the current version that I showed you is based on RI value only, and it's based on rules. So when you make some rules based on process, it can analyze based on the rules. And you know that the former version, which is 1.3, it has some rule-based analysis as well as AI-based analysis. And we will incorporate those features on the next version or in next coming year. But at the moment, it is not available. So at the moment, you will lose when you upgrade the software to 1.7, but soon you will have the updated version which incorporates the AI features as well. But the thing that you should be aware of is that it's about the important factor for AI utilization is the training. So in the new version, we'll incorporate the training workflow as well to have your own your tailored or your specialized version of models for your own analysis. So that will be different. So yeah, this was the question. I think all the questions are answered. So from now on, I'll show the real workflows on the TomoAnalysis. So this is how the TomoAnalysis software looks like. And at first, I show how TomoAnalysis starts. So at the